Welcome to another episode of Transformation in Trials. Today we're going to focus on bringing the cell and gene therapy sector forward. And in the studio with me, I have Alan Griffith. And Alan, maybe you could tell us a little bit more about what you're currently up to.
Yeah, hi Ivana. Nice to join you. Thanks for the opportunity to speak. I'm now head of operations, global operations at a company called Vector Builder. We are end-to-end, fully serviced CDMO. We've been in operation for about kind of 10 years now, with 4 or 5 years of that being as part of the CDMO. So yeah, heading up the operations there currently, but yeah, many years experience in cell and gene therapy, been in various clinical trials, exposure to clinical trials. So very happy to have a good discussion today.
Very excited for having you here. And we'll get more into some of your great experiences later on in the episode. But for now, because a lot of what we'll be talking about is cell and gene therapy, could you give our listeners some of the context for what is a vector in cell and gene therapy?
Yeah, sure. A couple of different ways to answer this, but and I'll try and interlude between layman's terms and scientific technical terms as well to kind of accommodate any listeners. But yeah, vector is basically any vehicle used to deliver genetic material into target cells. So if you just think of it as a molecular courier, I suppose, or in other words, a vector ferries genetic material into animal, human cells with the idea that these genes then can cause an effect, maybe replenish a gene replacement. There's CRISPRs now being put into these vectors as well that can also do gene editing. Basically, it's just a vehicle to get in, and they're the ultimate vehicle we've seen over the last few years for cell and gene therapy. They work really, really well. They come with challenges, of course, like manufacturing, which I'm sure we'll touch on during this
Yes.
conversation. But that's it. Yeah, in layman's terms, they're a delivery tool, taking the same idea as vaccines, but they're just empty and they're harmless, and we're putting good stuff back in them in order to execute good stuff in a therapeutic sense.
That's a great explanation. And now I'm seeing like a little fairy inside our bodies.
Yeah, exactly. And obviously the whole field has been quite good because due to the COVID response, of course, an mRNA, and I think a big portion are attributable to the COVID response and how fast it was. We have a lot of people talking to us about how fast it was and what happened there. A lot of people don't realize because of the state of play of the cell and gene therapy field, it really allowed not a cold start, but a reason to go straight in and actually utilize these vectors. The Oxford Biomedical one, for example, being a ChAdOx, which is an adenovirus, kind of ready to go. It's been tested at a small scale in research and development, but you didn't have to start from scratch. So I think that's something that's nice to align, that the COVID response was very fast due to the really good state of play of cell and gene as well, among other things, of course. But that's always good, worth remembering.
Yes, absolutely. Well, I do want to talk more about being a CDMO in the cell and gene therapy space. It's not an easy space to do manufacturing in. Can you tell us more about the circumstances for this?
Yeah, yeah, sure. Having been a developer myself for the last 10, 12 years, I've really kind of changed my role here now where, you're trying to generate money and get money up from various, investors, VCs, and going to board meetings to kind of beg for money, doing 2 or 3 projects and seeing a lot of failures, pitfalls, and challenges. But at a CDMO here now, me personally, I get to sit on and see hundreds of different client projects and utilize what I've learned in being a developer, as being a developer on payroll now, to actually help guide them on their transition or their journey from research, preclinical into clinical. But like basically in the CDMO sense, that just simply means manufacturing, doing the CMC side of things for these wonderful vectors. It's very much a case of people doing either buy or build kind of models where a lot of developers have the science. They have a fundamental decision to make very early. Do we buy our own facility and kit it out, repurpose it, and do our manufacturing ourselves, or do we outsource it to a third party? And that's where the contract developers come in. And there's many hundreds of them around the world. A lot of them older than us. We're a little bit later to the party, but I do think that's okay because some of the challenges that they've had at the early stage 10 years ago might be running out of obsolete equipment potentially, staff retention, the whole field itself is very volatile, so it's moving and changing daily and weekly. So by being a little bit later to the party, I think, Vector Builder in this case as a CDMO are a little bit at an advantage, which is great. But yeah, other challenges do exist because these products are so specialized to make,
Hmm.
and everyone thinks you select the CDMO, off you go and they'll manufacture it for you. But these things take so long to make and they're so expensive to make, all because the process equals the product in a lot of cases. Like that's something that's said now and again, that you don't have you have these platforms, but it's a bit of a misnomer. The platform wording is a bit of a misnomer. I get it. It's used by marketers all around the world.
Hmm.
It makes sense, but these therapies, they're so individual. The fundamental sequence and the vector sequence itself is so fundamental to the manufacturability of it overall. And this is something that makes sense now, but 10, 12 years ago, we were like, it can't be fully attributable to that. And it's not fully, but it's definitely a huge portion. So that's a huge challenge because if you get client X and client Y coming to you with the same gene of interest, we can nearly guarantee that there's going to be certain elements of the vector design that are different, even though the gene of interest remains the same for treating the same disease. Those discrete differences and nuances will make the vet manufacturing different, especially at scale. You go to 5 liter scale initially for feasibility, 20, 50 liter, and then obviously 200 liters where the majority of manufacturing is right now in the field. Obviously 2000 liters is possible, but there's very few people operating there and need to be there just now. But they're kind of the kind of challenges in the area. And just before I move on, I think one point I touched on the talent there, talent retention. That's a huge challenge in the whole field because making these things is quite specific, and you need a certain type of technician, scientist, postdoc, whatever coming into the businesses to manufacture and having exposure making them. But not many people have been making them for too long, so
Oh,
it's very hard. It's very hard to get the talent, and thankfully at Vector Builder, our model, because we are global and then we do a lot of the manufacturing in Asia. I think our staff retention is up around 94%, and that really has a compounding effect every year because you're learning on top of that. And when people say that brain drain can happen, thankfully here it's not happening. And I think that's just one challenge that we've overcome kind of silently. And we do like telling clients this as well because I think it does make a big difference when you're trying to deliver first time on these things.
in my experience, having like experienced staff for a prolonged duration when it comes to science is key to success.
Yeah.
If there's too much turnover, you start from scratch too much, and a lot about of it is institutional knowledge, like the know-how of making something work. So I understand what you're talking about. It's
Yeah.
crucial.
And I'm glad you resonated on that part because I know we talked about a good few things there, but the challenge is there, someone said it before, it was like working with a CDMO is kind of like with dancing with someone you don't know.
Yep.
And like, 'cause it's quite intimate, you have to partner with them, you have to trust a stranger kind of thing. So what we're trying to do here, for example, is not be very transactional, as in here's a product, please make it. It's like we want to really understand the disease. We want to understand what's inside it, what's the vector composition, what's the underlying sequence. That really makes the transition between tech transfer into manufacturing and all that so much easier. The risk is there. It should be absorbed by both the CDMO and the sponsor. And I think that's, the transactional side of things should be getting removed where possible. You need to trust us for what we need to do, and we need to be better at fundamentally understanding the clients. And that's what we do. Hence why I've come to Vector Builder now as a developer. We've got one or two more guys as well coming through that can actually help with those conversations because, you can pay lip service to people all around the world, but if you don't actually if you haven't been there, done that, got the t-shirt, I mean, it's easy to just, talk is cheap, and that's, I think, it's going to be a big differentiator for CDMOs going forward. Yeah.
I'm curious, what is a good profile for hiring into the CDMO space? You mentioned like several levels of technicians, PhDs, postdocs. Like, Are there personal attributes? Are there scientific attributes that make you especially great at this space?
Really, really good question. Been asked this before. Um, yeah, I mean, like there is a bit of a problem, I think, and I think a lot of CDMOs will disagree with me here, nothing controversial, but a lot of people work in a CDMO, and I've been in working in a manufacturing group. I've managed a manufacturing team, um, and they kind of, the more qualified they are, the more they kind of get bored really quickly. So it's about that self-motivation is a big driver. Um, you tend to get, a lot more numeration sometimes by being in a CMC role than you would necessarily if you're, in a research role. But because that kind of comes with the territory of being a little bit more monotonous, it's a little bit more routine. You should be robotic in how you do things in order to comply to GMP compliance. All makes a lot of sense, but when you're that person in the room doing the very important step and you're really not trying to mess it up, that's a lot of pressure. And then some people love the pressure, but then they get very bored of the pressure if they've manufactured loads of batches. So it's
Mhm.
that kind of mentality of person that you look for that understands that rather
Yeah,
than just maybe hiring people and letting them figure that out themselves and then kind of crossing your fingers hoping they don't leave. It's better to address it up front and then make sure that the company can facilitate learning in a different way or doing different tasks or different unit operations that they can, fluff up their skill set, make sure that they don't get, refreshed. And because a refresh technician there makes a lot less mistakes. And one of the biggest challenges in batch manufacturing is the failure rate. You have,
yeah,
one or two gaps in the GMP process, and you have to, tank the entire batch. That's where a lot of the cost comes as well with these things
yes,
because you have to allow for that happening because they're so complex. There's so many individual steps.
yeah. That's a good point, and I like that you bring forth self motivation.
Yeah.
Uh,
Hmm.
because for me also, whenever I have been hiring, looking for someone who understands, well, what I would like to achieve in this role is so and so, not for myself, but like for the company, for the product.
Yeah.
They usually tend to last longer, and also
Yeah.
like
Yeah. Yeah.
build those
Mhm.
bridges to other departments, and improve the process.
Exactly,
An excellent point.
exactly. Yeah, because I think it's a given that people will have the skill set, they'll have the quality.
Yeah,
Goes back to one thing, the marketeers kind of try and get to do. I work with the marketeers sometimes because they're like, give us the insight, and you can't really market quality because
no,
quality should just be given.
yes,
Quality is just a given that you're at a GMP standard and that's the benchmark, and you don't deviate from that. So you can't really say, hey, our quality is better than this CDMO. Let's be
yeah,
honest, like it's just a given, it's standard. Where the differentiator comes from is from, as we said, the staff, making
yeah,
sure we invest in the right equipment, make sure that we are actually able to conduct the work in a faster and better way, and try and find, efficiencies in other ways. But quality is just not compromised. It's not a marketing term that you should use.
yes, yes. But getting to that quality is actually not an easy thing to do.
No. No, it's really not. It's difficult, and it's, a lot of clinical trials right now are on, clinical hold because not necessarily always quality events, but the quality events that have manifested by maybe the underlying vector sequence or the vector design being putting the specification out of kilter or skewing it so much so that you do have a quality event, but that's the quality event against maybe the specification, not necessarily a safety quality event or an immunity. It's not dangerous to the patient, but you've still set a certain specification and then you haven't hit that. So
Yeah.
therefore, you have a deviation and you have to address that going forward. So that's an important point as well. Yeah.
Well, Alan, now you have had the luxury of observing like different kinds of companies, their vectors. I'm curious, how much do they have in common, and how much is different? You as a CDMO, like how much knowledge base do you get to build, seeing different products being manufactured?
Yeah, yeah, yeah. It's a really good point. Look, there's commonalities. There's more things in common than there are differences between CDMOs, if I'm honest. There's going to be regulatory requirements, robust quality control, scalability aspects, right equipment, the right people. But I think one of the aspects that people don't realize is quite fungible. I use the word fungible now because it's obviously quite in with the kids because NFTs and all of that. But that interchangeability or interconnectivity between these things, you know, it's no point having a quote from us and a quote from another CDMO and been like, oh, we're using some fancy equipment.
Mm,
Everyone uses a lot of the same suppliers, the same equipments, the Cytivas of this world, the Pauls of this world. There's not many equipment suppliers. And at the end of the day, you've got the top tier suppliers and you've got maybe top tier two. And the tier two are actually just as good in places, but everyone likes going for the more expensive units. I mean, that's what we've invested in. But we're not going to sit here and say our equipment is way better than CDMO X, because I think that's where, that's something that I think clients it's important for them to know because they're kind of hidden behind the veil of they don't know, they're not in their labs, they haven't got a window in. We try and show them, and instead of trying to say that we're better in certain ways, we just be very honest about that and say, look, where we don't worry about trying to look good with the equipment, we try and look good regarding the process and how we deliver the process and execute, because that's the USP for me. And then yeah, I just think sometimes there's a CDMOs that have a track record. That's amazing. Obviously, you've got into clinical trials, you've manufactured these things a lot of times. That's great. We're struggling in that regard, if I'm honest. We're not struggling, but we don't have the track record. We're a bit slower to the table, like we're a bit, late to the party. But that's okay because at the end of the day, we will naturally learn from what we're delivering. We're getting these products into patients now. Then, we're doing what I think is a lot faster and a lot more cost friendly, which hopefully we can touch on as well. The affordability factor is very important going forward. That hopefully we're starting to catch up with some of the big players or the pioneers in the field. And I think that's where I think that observation kind of comes from that, when I joined the CDMO, I actually thought that, I was a previous developer, client per se. And I'm like, oh, I wonder what equipment all these guys have. And the more I've seen over the last 2 or 3 years, everyone is pretty much the same thing. And anyone hears about a new disruptive technology coming, we'll always say, I kind of want that because I don't want to be either left behind or we're
mm.
starting to see publications or some of the key opinion leaders within the field now reporting on this equipment or reporting on these, machines to do assays and stuff. So that's quite important, I think, just to keep on top of it, keep the trends away, keep your antenna tuned in for what's kind of coming out there. And in that regard, it's pretty much a parity across the board, which is good. It
Yeah,
should be.
absolutely.
The client shouldn't be hoping they get the selection. The CDMO selection is a critical decision they have to make.
Yeah,
They don't want to sit there and go, God, I really hope I didn't get that wrong. It's like chances are, you should be okay, even if you go with somebody that doesn't, is not as good as you thought they would maybe necessarily be. And then final point on that, I just think, just to close that part, was I think the biopharma report from the Global Biopharma report in 2023. It was like 40% of developers are not happy with their CDMOs, but none of them really cite the equipment or anything. It's all
no,
about how the interpersonality is, how the
yes, yeah,
relationship, speed, like speed is not one thing, but the speed of conversation and going deeper into their project, listening to them, so that these are the things that the people, 2000 people, surveys and report. And that's quite telling. 40%, that's quite high, considering that there's a lot of people just not happy with their CDMOs. And then I'm hoping Vector Builder, we can maybe come in, and if people are not happy, they want to maybe test us out. Yes, we don't have the track record, but you're like, they're in a position now to make a change, and we want to maybe just be there for them because at the end of the day, we don't want them to slow down and getting these wonderful products to patients. Why would we?
absolutely. And again, like the human factor there in collaboration between sponsor and partner, also in my experience, is the most important part.
Yes. Yeah, exactly. It is. Yeah.
I'm curious. Well, so how what are some of the challenges of actually manufacturing these vectors? Because I assume like going from the development stage to actually being able to produce them, uh, that's is likely not an easy thing to do.
No, no, it's not. Yeah, it's a kind of stacked question. There's a few different ways I could answer it, so I'll try my best. But the way I look at it, something I touched on in the last answer was, that vector design element being so
Hmm.
pivotal to manufacturability. And, when we were going into clinical trials, when we, built up the business spinning out of UCL years ago, we didn't really know what sequences were clinically compliant, were acceptable. You have this huge knowledge now that ampicillin-based backbones are not the way forward. They are, there's probably not too much wrong with them, but there is this hyper like beta lactam hypersensitivity in the field that if you do manufacture a plasmid with ampicillin, there is a worry that there's trace elements of it coming through that could find its way into a patient. And I know that's a step or two above where the virus is manufactured. We need plasmid precursor material to make the virus, but you still have trace elements. And because it's hypersensitive, you might have that coming through. So we didn't know any of that in the first, 10, 12 years of making it. It was like we just didn't really care what backbone exists. We're the fastest way to clone it. It's in an ampicillin backbone. Let's just go. Whereas now, I'll guarantee you, there's not one developer over the last maybe 4 or 5 years would ever consider bringing an ampicillin-based backbone into the clinic, and even if it is upstream from manufacturing the virus, because
Hmm.
if you can change it to kanamycin, for example, why not change it at the very, very early stage, even when your researchers are coming up with the proof of concept data? Just get in the habit of doing that, and then you've got one less thing to worry about during your clinical translation. Because that could impact your manufacturing, it impact the quality control, the purities, all of that, empty versus full ratios, all of these things. So yeah, I don't want to harp on too much about the vector design, but I mean, there was a Nature paper there last week, Vivian Marks, and she had actually reached out to two or three people at Vector Builder and all the other CDMOs and people who are good vector, key opinion leaders. Everyone now is kind of coming around to the fact that, wow, this is really pivotal. It determines
Hmm.
therapeutic efficacy, it determines safety, it determines scalability. These discrete elements, a handful of bases, a handful of nucleotides within the sequence, can have profound effects as they get amplified up beyond certain scales. Um, we didn't even know that. Of course, 10 years ago, I always go back to the now versus then. So that's why you're saying, how we at least now can consult our clients coming through that here's two or three things that you can do now, as of
Yeah.
today, that will at least help you when it comes to manufacturing challenges later. A lot of the same challenges regarding chromatography development, process development, they still exist. But you've at least eradicated three or four big problems when it comes to compliance, safety, and scalability because of the way you've designed the vector. And that's a huge win. It's a huge win, and we didn't have that win 10 years ago. So I think that's important for people to understand as well, that the vector design, we talked about vectors earlier, it's that vector design instead of just maybe an off-the-shelf vector, you really take time scrutinizing it,
Yeah.
interrogating every single base within there. And we can do a lot of that work now as well. I think we're the only CDMO that, well, not the only CDMO, but as far as I know, the only CDMO at a certain our size that has the CRO part division of the business. So we've been a
Hmm.
very successful CRO company for years, and we've got a CDMO now. So we've got something like 400, it's 4,500 clients. I see that from our slides that there, even if a small percentage of those transition into clinical trials, that's still hundreds of potential clients that will come to us that need to transition their vector. So we're able to have that vector conversation with them early as they're ordering their research preps, as they're ordering their research materials, and they're like, "Oh, no, I'm not worried about clinical," and I'm like, well, we are like, yeah,
Yes.
when I know you're not, but that's a bit of a problem. Here, why not just make these two or three changes? We won't
Yeah.
charge you any differently for changing them, and then you'll be kind of ready. And that sounds like, we've been condescending, but we're not, because it really helps us as well. Because we don't want them to spend 2 years doing proof of concept and then they're like, haha, Eureka, I have the vector I want. Here, vector builder, can you please make it? And then we do the assessment, the vector assessment, takes an hour or two to look at it. And we're like, we already know after making a million
Yeah.
vectors at this stage that this is not manufacturable.
Yeah.
And you could have had that decision, and now you have to go back and maybe repeat some of the work. So that's the big, I think,
Yeah.
role that CDMOs and the knowledge transfer has regarding boosting manufacturing success, if that makes
Oh,
sense.
absolutely. Like bringing that expertise to the earlier stages of research, it
Yeah,
makes life much easier downstream.
big time. Yeah. And there's other companies looking at the in silico models where, you can look at the in silico side of the sequence, tertiary structure, snapback genomes, the formation of those. I mean, that's all great, but I mean, the AI and machine learning that's obviously coming at full pace, and we're trying to keep our, antenna aware of those. But there's an empirical part of that as well. We have we have huge process development labs and capabilities where we could do the small scale experiments at 1 liter, 5 liter, and then you are able to screen like a DOE type situation where you're doing loads of different iterations of the vector design. Because I think a lot of the AI tools that some of these companies now have set up, they're like, make a better vector design using our AI tool. I'm like, but have they got the capability to do the empirical testing? Because fundamentally, the AI tool, 9 times out of 10, it could be excellent, but then 1 time out of 10, it's not. And then you'll never, as a developer, be able to get that time back. That's a lost time. So parallel tracking it with the AI tool, but then doing some empirical work. That's part of our CliniVec service. It's a term I've coined working at Vector Builder, but CliniVec is a clinical compliant vector, build clinical compliant vector. And then it's based on the three pillars I've always mentioned: the manufacturing, the safety, and scalability. So that's hopefully we're starting to see people inquiring about that now, because the problem with it is it costs more money up front, whereas they might have said, we've only budgeted for one preclinical candidate, and you have to try and convince them, hey, you should really look at least 20, 30 preclinical candidates. I know that sounds crazy. I know that sounds like loads more work, but that's a really low cost burden at that early stage where you don't
Yes.
have GMP involved, you don't have large amounts of virus to be made. That's the time to really iterate, do iteration and really focus on it. The amount of clients that have said it makes total sense, but I've told my investors I need to pick one within a month, and you're like, well, you are putting yourself at a disadvantage there, but we're not going to tell those people to go away. Of course, we're going to try and help them as fast as we can. So
Yeah.
that's important. Yeah.
But that it's a good point about the scientific process in general. Like we have, it's cheaper to experiment earlier, and it gets more and more expensive the later you experiment.
Yeah, but unfortunately, we talk to them well after they've had the conversation with the investors
Ah,
and stuff,
yes,
you know.
yeah.
Yeah. But
Ah,
that's good. That's good because we've some research clients now, and they're like, "Hey, Alan, I work very closely with Andrew Alcock, their previous Oxford Biomedic gentleman, and they're asking us to attend some of their investor meetings because they're like, can you please just talk about all that manufacturing CMC stuff you're talking about and why it's important?" And they almost use us to convince the powers that be to actually spend more time up front and do better development. And we think that's great because
yeah.
building the relationship, it's allowing us, exposure in that realm, and it's like it's the right thing to do because
Yes.
there's no point just picking one needle in a haystack and then hoping for the best. To be honest, that's where a lot of CDMOs get a bad reputation because they actually haven't put that stage gate in place. They've kind of just said, yeah, you come to us and we'll manufacture it for you, and not appreciating the complexity of the underlying vector design. And then they're like, we can't deliver, we can't manufacture it. And then they're tainted forever that the CDMO can't make our bloody thing. And you're like, well, you have come about it the wrong way. So I'm not saying we're going to be totally avoiding failures, but we're at least giving ourselves a better shot and a better chance at success. And I think we're all quite excited about where it's going for the next few years with some of our big orders coming through for clinical trial orders.
That is exciting. I do want to circle back to the price part, because that's like the that's the thing in cell and gene therapy is the price.
Yeah.
How do you see that it could become more affordable?
Yeah, tough question, man. You weren't letting me away with that one. Yeah, look at it. At the end of the day, it's the $1 million question, or it's the multi-million-dollar question. Let's be honest. This question's been around as long as the field had that resurgence, 15, 20 years ago. I don't know the best way to answer it, but I'll try and think about a few different ways. Comes back to what I was saying. We have that Clinivex service there now. You
Mhm.
spend that money up front, and fundamentally, then you're not spending money on failed batches later. I'd rather spend 10K now than spend 100K on and repeating another batch. So we're trying to use Clinivex to really bring the affordability factor down. And okay, that sounds like we're spending more money up front, of course, but there is a payoff kind of later. So obviously, the affordability hinges on these kind of efficiencies. Other way to do it is collaboration. A lot of people were always very competitive right now. You're seeing a lot of people divesting, a lot of people collaborating and doing strategic partnerships with people to really not not do everything all under one roof, because by the time they develop it themselves and then roll it out, they've spent millions of pounds themselves and the client could be gone. So I think it's about leveraging who's available, who has what. If you're not really good at fill and finish, maybe considering outsourcing it until you become really competent at it. These are the type things that you can do, I think, from an operational perspective, and that's
Yeah.
kind of my bag, of course. But yeah, this question has been around the boardrooms for years. It's heavily nuanced, unfortunately, but um, kind of a simple example I always say to this is for the kind of lay people or anyone who's reading or listening, but 10K or 10,000 is obviously a dose for maybe small molecules would be considered very high, historically. Whereas 10K for a gene replacement therapy is considered very, very cheap, and someone
Mhm.
thinks you'll have to get your head checked. So there you go. We're trying to be like small molecules was, in MABs, 20, 30 years ago. We're trying to be the same in that, work the same way, get us up to that kind of level. But that price is probably never going to be really achievable. Maybe for certain diseases where the dose requirement's a lot less, fair enough. But
Mhm.
all things being equal, we just have to kind of accept that it is really expensive. And then the fact that they are um, hopefully going to be more life-changing, better, more long-term. There's a huge durability factor. It's a bit of a misnomer. I think I'd like to come back to some of the misnomers maybe towards the end of the podcast, but one and done, people say you treat it once and then that's it. We were hoping that was the case 10 years ago, but time has shown now that there is a kind of a tailing off of
Mhm.
expression, and this durability factor is not really quite there. So that was always going to be our angle to say, hey, it's expensive, but it's a one and done.
Yeah.
That's now coming back into focus where only in
Right.
certain
Yeah.
circumstances, only in certain cases
Yeah.
that holds true. So that'll be interesting now to see how everyone in the field manages the economics around that.
Mm.
Hopefully that makes sense. Yeah.
Yes, absolutely. I do want to hear more about the misnomers.
Oh, really? Right now, you want to hear? This is the thing as well. I was hoping I'll try and choose two or three that I think people will agree with me, because I think at the end of the day, here we're talking and I'm trying to be not as subjective as possible. Everyone has a subjective experience, but
Yes.
I'm really just trying to talk about, what is misnomers. I think in the field, they can really misalign expectations, because for that one thing, talking to investors, one and done, one and done, if you actually explain to them what the way I've done it there, that okay, now we know 10 years later, we've learned from all the clinical
Yes.
trials ongoing. There's a public misunderstanding in place, so therefore we want to address that with you as well. So you have to try and, as the field advances, look at, better education, better clinical data, and understanding this data is really important. So that's a misnomer I think is very important. Another one is the more vector there is, the better efficacy you have, as in more vector, more this thing cures you. The more of it you have in place, the better off you'll be. Now, higher vector doses, the way they work, does not always mean better results.
Mm.
Excessive vectors sometimes can lead then to immune responses, toxicity, off-target effects. These are three big issues in the entire field. So more is not better. And you can use that in many circumstances. So yeah, I just think that one-time treatment is something that is there. Again, some investors will probably kill me for saying it, but I think it's just that's it's our responsibility to make sure that it's very holistic, the understanding
Yes.
around that, and yeah, removing some of these misnomers is totally fine, because I think they can be replaced with better solutions. It's not a case of, oh, we're all screwed. It's not about that. It's just about, we know stuff now that we didn't know then, and let's remove some of these misnomers, because I think that will help avoid misaligned expectations, and that's our clients as well. The clients come to us and say, yeah, but I was talking to someone else and they said I could do it in 50 liters. They're like, what design is it? I haven't talked to them about the design yet. Okay, for them to tell you that you're 100% going to get 10E13 in a 50-liter bioreactor without knowing your vector design or your
Yeah.
underlying sequence, well, honestly, they're just trying to lure you in. Let's be
Yeah.
honest. It's a good sales tactic. It works. And let's be honest. There's a 50% chance he's right, or he or she is right, and 50% chance they're wrong. We're just really trying to tackle it from a realistic perspective, that you can't say that and be 100% true. And that's okay. We're here to help with that understanding.
Yeah. Absolutely. Uh, and I think those were great points. And also, this is just the scientific process. We learn more, and then some of the things we're assuming in the past no longer hold true, and then we need to adjust
Exactly.
our expectations.
Yeah, exactly. Every idea is a bad idea until it's a good idea, you know.
Exactly. Yeah.
Yeah. Yeah.
Eventually, we'll figure out the price one too, with
Yeah,
given
yeah,
enough
yeah. It is going to be
experimentation.
interesting to see, people are focusing on routes of administration, so you try more, more targeted route of administration, because if you go more controlled and less systemic, you can bring that dose way down because it'll be compartmentalized in a certain tissue. Like the brain is great for that due to the blood-brain barrier. The eye was obviously great because it's got, immune privileged, and it's a perfect control. You've got a left and right control. That's why a lot of ocular gene therapy companies, ourselves included, really set up at the start, because it was a really good way to keep the dose down. The manufacturing is quite, mediocre, as in the cost, and allows you to kind of get that familiarity, get that experience, get that competency, get the investors excited, and then you can maybe diversify your portfolio. Look at CNS diseases, look at other therapy or other disease pathologies as well. So that's kind of important. If you're going into cell and gene therapy, manufacturing is not your only challenge. You need to actually understand the disease inside out. Whereas the problem, I think, in the field, a lot of cell and gene therapy experts, they're not pathologists, they're not clinicians, so you really have to help with that conversation early. And I honestly think if you have those better conversations early, it can probably save you a lot of time, money, and effort later on. And that's something that kind of makes sense, but I don't think the systems or the infrastructure in the field are really set up for that. It's getting better. There's biofarm, there's groups that are set up. There's lobbyists now, and there's all of these things coming together quite nicely to kind of create these systems or these infrastructures that can allow the right people, to have the right conversation early as possible. That's going to be fundamental to the cost coming down as well. Sorry, not to go back on that, but you touched on it again there, and I was just continuing the same thought process. Yeah.
It's an interesting point. We talked a lot about like the initial stages when it comes to research and manufacturing. What about the regulatory approvals for cell and gene therapies? What are some of the challenges and developments we're seeing in that space?
Yeah, yeah, great. This is a great question because we're talking a lot about manufacturing, talking about a lot about cost, but the regulatory landscape has changed hugely over the last decade as well. The agencies, they're all learning pretty much the same as these developers and even the CDMOs. You try and my recommendation there is, don't assume what you heard in a maybe from an agency meeting 5 years ago. Things could have changed there. It's really good at a lot of our cell and gene therapy conferences, the likes of Peter Marks from the FDA and all of these kind of speakers and good communicators. They're not treating it like a black box. They're actually getting in and amongst the people and trying to tell us that, hey, look, we thought this, it's changing. We weren't usually worried about this. Now we're starting to be worried, as in like AAV integration, for example. We always like, no, AAV doesn't integrate. That's one of the challenges of it. It remains episomally. But at the same time, now we're starting to see that, no, it actually does integrate at low frequency, fair enough, in certain parts of the host genome. So the question then is that okay? Is that bad? Is that good? Is
Yeah.
that, dangerous? And then like the agencies are like, you tell us, like if you think it's safe, show us and demonstrate it that it's safe. So I think that's what I've learned a lot from the regulatory kind of landscape over the last few years, that it's changed. And be comfortable with being uncomfortable a little bit. I'm trying not to use too many cliches, but a lot of people think they're trying to catch us out, but they're not. They really want you to succeed because there's room at the
Yes.
table for everybody, and they want to, like any human being, they want to do less work. They want to be able to set up these kind of more like conventional frameworks that allows people to move through the clinical phases much faster. They get no advantage by it being slower. It's just
No,
giving them
no one
extra work,
does.
you
Yeah.
know? Mm.
No, absolutely. Um, well, during our pre-call, you mentioned that it's been a difficult year for the cell and gene therapy space last year. Can you elaborate more on what made it a difficult year?
Yeah, yeah, sure. Few different ways. I suppose the challenging year is obviously, the investment money was drying up a lot. I think a lot of people were cash rich over the last few years, and a bit of on the boom. We're starting to see a bit of a recoil naturally because, as I said before, the clinical trials, some of them are on hold, and they're on hold without really a clear way forward. As in, you don't necessarily mind a clinical hold now and again because you've found the root cause, you know how to do it. the only pain within the entire business is that it's delayed everyone by 3 to 6 months, but you actually have an answer, you have a way forward. Some of these now clinical trials, they don't actually have a way forward, so they're having to go back to the drawing board and actually think about things a bit deeply. So that clearly translates into investor sentiment and what's actually like, these guys are great scientists, but they're not really great at developing these things into scalable manufacturing modalities. And when you think about it, it's not it's a criticism, but it's not a criticism at an emotional level or a personal level. It's like, I think the investors are just wising up to the fact that, oh, well, it's a lot more trickier than we thought. Just because we had 2 or 3 success stories, as I said, in a certain disease area that could be quite applicable or amenable, sorry, to certain gene therapy, it kind of biases everyone else's opinion that, hey, no, gene therapy is easy, and I'm sure in some cases it is. So I think that's then been the reason for the challenging year, that the money's dried up. But having been at JP Morgan this year, there is kind of now that resurgence again, the conversations happening. We're talking about things like the Clinivex, like the equipment, disruptive technology users are finally catching up with us. Um, Quality control groups and quality control companies and the guys that manufacture these wonderful machines, they're starting to really come into their own now, and that means better characterization of the virus earlier in the development life cycle. And then that means you can make way better decisions earlier without going and hitting those clinical trial roadblocks. So I think the investors are also aware of that, oh, it's not just a dead rubber. The whole field hasn't come to an abrupt stop. Yes, it's at a bit of a plateau, but there's a bunch of smart people trying to figure out ways to counteract the problems that may arise in clinical trials, manufacturing just being one of them. So yeah, hopefully that makes sense. And then I think,
Yes.
yeah, the orders, you can see it translate. The orders just come down low for the last year or two. And again, look, at the end of the day, a lot of these profile projects have failed. But there's always going to be either new products coming into development, or I actually think there'll be a huge resurrection situation where, you know, 1 or 2 companies can get to clinical trials, they meet a roadblock, and they realize we made a big boo-boo here. We missed the point here that could have saved us this hassle. But unfortunately, we need to go all the way back to phase
Yeah.
one and repeat the work. I actually think what may happen is you'll get a lot of developers or people coming in, or maybe, like the biosimilars type mentality, where they come in and say, the only thing that failed, that caused that wonderful drug or therapeutic to fail, was X. We can fix that now, and we've the money to bring that all the way through. So you might see these resurrection campaigns or these resurrection companies setting up where we'll buy the rights for that project off you, or that indication
Yeah.
off you, and go forward. And I think that's still the great white hope of like the investor money. That's good bang for your buck. That's where confidence is starting to creep up. Yeah,
Yeah, and because it's still a relatively new space, we do need those examples where there we go. We've actually figured out what was wrong, and now we can do it all the way. Uh,
exactly,
that
exactly.
that'll be
Yeah,
helpful.
yeah, yeah.
Well, Alan, I would like to learn more about your journey into this space. How
Okay.
did you end up in the cell and gene therapy area?
Yeah, sure. Went to the shop one day and then
Bought some
no,
eggs.
yeah, I actually fell out of love with science big time after my PhD. I was doing it with a wonderful PI and a wonderful organization, the National Institute of Cellular Biotechnology in Dublin City University. However, it was just kind of partially funded in different ways by Big Pharma. I wasn't allowed to really talk too much about what some of the findings were. I felt like I was doing a lot of work, kind of maybe that'll just get put in a filing cabinet and logged away and not actually go anywhere. So I wasn't really too sure what I wanted to do next. All I knew is I had viral genetics background. I done bioprocessing, phenotype mapping for my PhD, which is basically utilizing viral genetics. So I wasn't really necessarily in gene and cell therapy per se. It was a lot of the same vectors and materials that's used in cell and gene, but I wasn't actually in the cell and gene gene therapy sector after my PhD. I decided to go to UCL and apply for a role there, working on Professor Robin Allen Ali's group, very successful professor there who built out and spun out a company called Athena Vision, which went on to be Mira GTX out of UCL. And that was a hell of a journey because I was like the fourth employee there. We literally built up the company within a few years. We built our own manufacturing. Again, would
Wow.
we do that again? I'm not sure. But we definitely decided to build our own and control our own destiny kind of thing. But that's pretty much what happened. The rest is history, as they say. I went up to director level there, then went to become vice president of research at another startup AAV company in London. Went to a short stint in the Nordics to work on adenoviruses as chief development officer there. And subsequently, yeah, money kind of runs out and decisions, and you start reflecting then over the last 2 years where am I okay, begging board members and investors for money just to do this work that I don't, they're asking me, are you sure it's going to work? I'm like, I really mean the team. We don't bloody know.
It's
But
research.
yeah. So that stress kind of takes its toll, and
Yeah.
obviously trying to start a family and stuff. And then I decided I used Vector Builder for many years as a useful service provider. Had the right conversation with the chief operating officer there at a conference, told him pretty much the exact story I told you now. And he was like, come here, I think there's a job here we can work on this, and you being on like a developer on payroll would really help facilitate. So I consulted for a few months, and then they've asked me to join full time as head of operations now, and it's great. It's a global company. I get to work in China, I get to work in Asia, Japan, America, all of that. We've sites all over the world. So that's kind of how I'm in front of you today, because obviously that connection there, and obviously your good relationship with you already. So yeah, but the journey was driven by passion for getting into patients, because I think this was the new area. This was an emerging area of biotech that was growing 10, this is 12 years ago, that postdoc. And then you're like, I was a postdoc for a few months and decided I want to work in this startup company. I can't pay you anymore, Alan, but I do want to stay, and let's see where it gets us. And here we are, 12 years later. And it's been a hell of a journey, and it's great. I'm so proud to be part of it. I'm delighted how it was. And there's so many learnings as well, which probably won't go into, but there's a lot of things I do differently. There's decisions we made, and you're like, I can't believe we wasted so much money doing an NHP study when it was the wrong thing to do. But that's why I'm here now. I'm here so
Yes.
I can hopefully tell at least if one or two of our clients listen to the same kind of pitfalls that we had, that's good use of my time, because that's really helping bring them forward, bringing the
Yes.
field forward, and reducing the valley of debt. The valley of debt, a lot of products go in there, none of them come out the back end. If we can reduce the value of debt, I'll be super happy. And that's what drives me forward. That's the passion. Yeah.
I can completely relate to that. That's
Mm,
but also like you got so many different perspectives on this field from different areas.
yeah.
That makes you an amazing partner for
Can you
any
tell my
client.
wife that?
You can quote me.
Yeah, no, it's good.
um,
Appreciate it. But yeah, no, it's good. It's been a good journey. I'm lucky. I'm looking forward to the future. At the end of the day, there's none of this. I think the stagnancy, even at last year, was quite challenging, as we touched on. Like, it still has the ability to go this way. And now you've got
yeah,
non-viral gene therapy coming into its own. We've just invested in a brand new mRNA LNP facility, LNPs, exosomes, these non-viral vectors. So a vector, again, we're talking about, it's a vehicle. There's now non-viral vectors, which always existed, but they're really coming into their own over the last 2 or 3 years. So it'd be interesting to see how viral vector versus non-viral vector, how they stack up against each other. Viruses were I wouldn't even, a lot of people say I never use anything other than a virus. But we're seeing now that some of these viruses don't like being manufactured, regardless of how you design them as well. So
yeah,
that's another thing. Just because we design them well, a vector builder doesn't mean they're 100% going to work. And some of these things just don't like being made, because you have to make them in these mammalian systems or bacterial systems.
mhm.
And then you're putting viral pieces inside a bacteria, and the bacteria is like, hey, I don't like what you're trying to
I don't
do
want
here.
it. Yeah,
Yeah. Yeah.
that's interesting. It's definitely a field that I'm interested in keeping an eye out for
Yeah.
and seeing how it evolves.
Yeah, it'd be
Well,
exciting. Yeah, yeah, and
yeah,
the money. I think we talked on about the investors a lot there. Didn't actually mean to do that, but I think they're quite interested as well, because that cost and affordability, viral vectors will always have a place because they work so well. They're not going to be eradicated or become obsolete. But it's just a case of some of these new developers coming through with these new indications or disease targeting therapies. There is a chance that the cost could come way down because they're a lot easier to synthetically make. You're not waiting around for the virus to do its assembly process. It's a very natural phenomenon that happens. If you can just synthesize them using chemistry and using, there's 3D printing of molecules going on. I mean, that could bring the costs kind of way down. And then maybe we find ourselves in this place where the majority of people are using non-viral. However, I firmly believe that they will be using viral vectors for the next 100 years because they're just too good in certain diseases in certain contexts that they couldn't be eradicated. So we need to keep fighting on both fronts, basically. Yeah.
Yes, and that well, this episode turned out to be excellent investor advice because it's both about looking out for the people who have failed and then trying again, invest in those,
Yeah,
and also don't forget about the viral vectors.
exactly. Exactly. Exactly.
Well, Ellen, as we start rounding off, we always ask our guests the same question in the end. If I was to give you the transformation trials magic wand that can
Yep.
change one thing in the life sciences industry, what would you wish to change?
Well, yeah. So I'll try not to be greedy here because I was like, I had I seen your previous episode and I knew that might be a question here. But obviously the cost is, I think that just goes without saying. Everyone wants the cost to come down. But my magic wand, I'll save it for I think more accessible QC that is
Mm,
harmonized because the quality control is always lagging behind. And I don't think this is the number one thing that needs to change. It's just if I had my magic wand,
yeah,
much pain quality control and characterizing our viruses while making them has put the CMC groups and the manufacturing groups in a disadvantage in going into, regulatory meetings. You get asked what's going on here, and you're like, it's this QC method is the first time it's ever been used to characterize this virus. Whereas now a lot more data out there. Some of these viruses, not only do they not like being manufactured, they don't like being tested and assayed and assessed. So I kind of feel like I'd love that where everyone, all CDMOs, all clients are all working in a similar kind of fashion that you know, that harmonization of quality control. I'd really advocate for that because I think enhancing that will really help not only us deliver manufacturing and all of the things I've said, but also the regulatory side of things, which is
yeah,
back to what we're saying. It makes regulatory guys assess the information so much easier because they're like, whoa, everyone's using more or less the same assays here. There's like 16, 17 release assays. We're seeing a lot of them using the same. This is great because now we're starting to see, structures forming, trends forming. We can trend the data. I think that's quite important going forward. But obviously, there's other things that we change, but that's what I'd really find that would really help us right now and the field. Yeah.
I love your response. Also because this is something we can actually do something about.
Yeah, 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 exactly. Yeah, yeah. And it's very doable because a lot of people think of the QC side of things a little bit as an afterthought, and they have. I think it's getting better now. But as I said, CMC, the second C in C stands for controls, and controls means QC. Everyone thinks CMC is manufacturing, but it's like, no, it's the quality control built into the manufacturing, and they have to go together. Unfortunately, over the last decade, the manufacturing has improved a hell of a lot, and
Yeah.
QC is kind of stayed around the same. That needs to change. We're doing a lot of work on that in Vector Builder, and I'm sure other CDMOs are as well. But yeah, I think that's a big focus for us now going into our newer initiatives around, you have to spend that money in that area, and you're kind of like, what if the client is very happy to use a very older kind of assay or methodology, and you're like, we've just spent millions developing these wonderful QC, but they're not ready to hear the conversation yet. That's a risk, but
Yeah.
it's someone needs to make the step. Someone needs to take the step, and we are trying to do it. And we're not going to go alone. We're trying to align with certain collaborators like Testavec and these companies in the UK. Pebble Biosystems, they've got these wonderful organs on a rig that you can actually dose the AAVs, for example, on it, and you get like liver and kidney readouts without actually having to go into animals. So therefore, your development costs come way down because you're not spending money on animals, and it's also helping the three R's, refine,
Yes.
replace, and replace. So I think that's very important as well. There's an aspect of that we're working on, and we're trying to be innovative in that space rather than just keeping the status quo.
That's really cool. Also, like for actually scaling the manufacturing,
Yeah,
uh, getting the QC right from the beginning. I bet that also makes a difference there.
exactly.
I love your answer there. Thank you.
No worries. Yeah, yeah.
Uh, well, Ellen, if my listeners have follow-up questions and want to get in touch with you or Vector Builder, where should they reach out?
Yeah, sure. I'll obviously I'm at Vector Builder, but get me on LinkedIn or whatever. Very happy. As I said, I've consulted before as well. Generally, I'm not a consultant that charges people. I like just helping the field because I'd want people, because generally by having a conversation with someone, it works both ways. I end up gathering information or learning something from them as well. And that's how we kind of need to work a little bit more as a collaborative, like co-creation is something that kind of gets mentioned a lot as well. But that mentality shift I think is important now. And it used to be very tight-lipped and IP protection, and I get all of that, but there hasn't been many people fighting each other on IP and stuff in the cell and gene therapy space, as a when you think of biosimilars and you think of those other areas. So I think we should actually just advocate for that a little bit more together.
Yeah,
But yeah, but if anyone wants to reach out to me and have any discussion on anything I've talked about, free for a conversation. Love to have a coffee, whatever. But yeah, LinkedIn, my Vector Builder email, I give it to you as well, Ivana. You can get grab me there. So yeah,
awesome. Well, thank you so much, Alan. This has been a great conversation.
yeah, amazing. Thanks for having me, Ivana, and really, really, really enjoyed it. Thanks very much.